Okay. This is like a missing link slideshow. Lost episode. I don't know. I don't know why I didn't record it last year, but I don't have a video for this section, so we're gonna record it this year. Topic is non-Mendelian inheritance. So basically, everything we've done up to this point was at least variations of Mendelian inheritance. Looking at just how traits are passed down in pedigrees, looking at Punnett squares and how traditional inheritance can work. We'll have looked at linked genes and how we can tell if the inheritance patterns are seeming non-Mendelian. And this is really just going to go through like all of the main scenarios that we will get non-Mendelian inheritance. And when I say non-Mendelian, I mean outside of the realm of just two copies of every gene. Well, actually, why do we go? What do I mean by Mendelian? So Mendelian inheritance is two alleles for every trait, only two copies basically, in two different versions, and that they separate. We now know that they separate during the formation of the sperm and egg, and they end up in different gametes. Mendel was basically just saying you have two copies of every trait. At some point in the reproductive process, they split, and only one of them ends up actually getting passed down. And then his other law was the law of independent assortment, which basically just says that there's no rhyme or reason to which cell each of the allele pairs go to. So if we imagine like the mom and the father, we can say mom's blue, father's red. All the law of independent assortment is saying is that there's no way to predict if you'll have like all blue in one cell or all red in one cell. Will it be a mix? It's basically just saying it's random. It's not like it's putting all of the maternal chromosomes in one cell and all of the paternal chromosomes in another. It's just completely random. You do need to know these two laws. I recommend just memorizing them by name so you don't have to describe what they actually mean. And the importance of these is really for genetic variation, independent assortment. Again, the orienting randomly during metaphase 1, meaning we have a mixture of chromosomes from mom and dad because we have 23 possible chromosomes, and there are two different versions. We have 2 to the 23rd power possible combinations just of different chromosome combinations. Then we get random fertilization, which is the idea that any sperm can fuse with any egg. So we have millions and millions and millions of sperm, far fewer eggs, but the genetics in each of those is random, and they will randomly combine. So overall, if we combine those two, we end up with about, for any one reproductive event, so like every child, there's about 70 trillion different possibilities and combinations, which is a mind-boggling number. This does not include crossing over. With crossing over, it's probably uncalculable. It's just a crazy, huge number. So we get basically infinite different combinations. So, non-Mendelian genetics, the main counterpoints to his theories is that one, and we're going to talk about these in more detail, one, some alleles are not actually completely dominant or completely recessive to each other. Some genes have more than two alleles, so more than two different versions. There are more than two hair colors, for example. Some genes can actually produce multiple different phenotypes. And then some genes are only passed down by the mother. So let's talk about degrees of dominance first. So Mendel had complete dominance in his pea plants. He was kind of lucky. Well, partially lucky, partially he did still pick those traits specifically. But all this means is that the heterozygote and the dominant homozygote are the same. So it doesn't matter if you have, like for brown eyes, it doesn't matter if you have one blue eye allele. As long as you have one brown eye allele, you have brown eyes. And you wouldn't be able to tell versus someone with two brown eye alleles. They just look the same. Incomplete dominance is when the hybrid, the heterozygote, is somewhere in between the phenotype of the two parents, which we can see here with the roses, flowers, whatever they are. If we cross a red and a white flower, we do get red and white flowers, but the heterozygote, the one that has one red and one white allele, ends up being a pink flower, which is just between red and white. The last type is codominance. This is when essentially both alleles are dominant, and they will both show up in the phenotype in separate, distinguishable ways. So it's not like a blending of the two. It's like they're both there. Kind of like, think like polka dots almost. The human example is blood type. You can be type A, you can be type B, you can also be type AB. We'll do more with this. We will take our blood type in February. Some common misconceptions. Dominant alleles versus recessive. Dominant alleles don't like subdue or cancel or destroy the recessive alleles. They just generally code for a thing to be there. So like coding for a pigment versus not coding for a pigment being not coding for a pigment being the recessive version. Also, dominant alleles does not mean they're more common than a recessive allele. For example, the trait for an extra finger or toe is a dominant gene, and it's 1 in 400. So you only need one copy, but that doesn't mean it's a very common thing to have. Again, talking with multiple alleles, this also goes with codominance. Most genes have more than 2 forms. Think of all the different possible hair colors, eye colors, heights, skin tones. Like, most genes don't just exist in 2 forms. Most have multiple. And you can see the codominance of the AB here, where A is triangles on the outside, B is circles, AB is both triangles and circles. So they're both showing up. It's not like a round triangle that is showing up. It's just both. Next, pleiotropy. Most genes don't do just one thing. Remember, we have 22,000 genes, and wherever it can, evolution likes to reuse its old work. So generally, we will have one gene that will have a lot of different phenotype effects, a lot of different symptoms, you might say. So like the gene for sickle cell anemia can lead to blindness, liver failure, heart attack, all multiple different symptoms, traits, but it arises from one gene. Okay. Next, epistasis and polygenic inheritance. Polygenic inheritance is the more common one to need to know. It's just that the genes have an additive effect. So skin tone is an example here. In this example, there are 6 different pigment genes, melanin-producing genes, essentially. And the darkness of your skin tone is basically just determined by how many melanin-producing genes you have. So if you have none, you're very pale white-skinned. If you have all of them, you have darker skin. If you have 3, you're somewhere in the middle. But it's kind of like a gradual gradient. It's a smooth transition. Epistasis is different. It's basically where one gene can affect the expression of another gene. So we see this with the fur of Labradors, where they have one gene that basically determines like the color of the pigment that they're going to make, and then another one determines if that pigment is going to be deposited or not, or like actually put into the fur. And so if they're lacking, which the E one I believe is the depositing in the fur gene. So if they don't have any genes to deposit the pigment in the fur, you end up with a yellow lab no matter what melanin producing genes they have. And then you get chocolate labs, red labs, etc. But you have 2 genes kind of working together to produce a single phenotype, a single look. So the dogs have one for what color of fur they're gonna make, variations of brown or red. And then they have another one that is, do we actually put that pigment into the fur? If we don't have that, then it becomes a yellow lab. We also have genes that are affected by the environment. So, for example, hydrangea flowers, the color will depend on the soil acidity. So it's the same gene, but you get two different color flowers. Certain lizards, the biological sex actually gets affected by the temperature the eggs are incubated at. If you think of rabbits in the winter, they go from like browner, kind of brown gray coloring in the summer months to white in winter. It's the same gene, but it is producing two different phenotypes. And then, almost lastly, two more. We have phenotypic plasticity, which is, again, one genotype, multiple possible phenotypes. This really does relate with environmental effect. This is just more with another example, basically. So it is just a change in, do we actually make the protein the gene codes for? So an example, tadpoles, I already talked about the rabbits, tadpoles, they can actually have different mouths depending on what food is present in the ponds that they're living in. So they can have omnivore morphs versus carnivore morphs. If there's plant life, they'll be an omnivore. If there's not really many plants, then it will become a carnivore morph. But same genes, and both will become a frog that you wouldn't be able to tell. Lastly, we have mitochondrial genes. I will have probably mentioned this as we go through pedigrees, but just as a reminder, all mitochondria are inherited from your mother. So if it is a mitochondrial disease, if your mother has the disease, all of the children will also have that disease, because all of our mitochondria come from our mother. So they show different inheritance patterns. They show up different in pedigrees, which, as you can see, there's pedigrees. Did it in a slightly different order this year. I wanted to spend more time on the processes and less on this memorization stuff. And I kind of had it flipped last year, so that is why. And that is all. Farewell. Good luck. I'll give you a point of extra credit if you email, I'll say, not if you, to the first 5 people that email me the word kumquat, like the fruit. Bye.